Good morning, everyone. And first and foremost, I would like to welcome you to the 2023 Translational Research Acceleration Program Proposers Day. We will just wait for a few minutes as everyone joins the webinar. So we will be back, and let's give it 1 minute, and we will get things kicked off. If you're just joining, we're giving a few minutes to let everyone join the webinar before we get started. We'll get started here in a few seconds. Well, good day, everyone. Again, my name is Chad Jackson. I'm the Senior Director of the Preclinical Translational Research Program at the Foundation Fighting Blindness. And we're all here today to discuss and talk about the Translational Research Acceleration Program Award. I really appreciate everyone's time and attention to this because it's such an important funding mechanisms that are geared towards bringing solutions to those with inherited retinal disease and dry AMD. So just a little bit of the overview of the meeting's agenda. For about 30 minutes, we'll discuss the TRAP overview. This is just highlighting the major components of the TRAP award. Then it will be followed by just a general discussion around some grant writing tips. I really want to highlight this as, I want everyone, when they're submitting either the letter of intent or full application, if you're invited, to really pay attention to grantsmanship, as it's very important and helps really easily convey the message to the audience. And lastly, we'll finish up with some time with questions from the audience to provide any clarity possible that we can for the TRAP award. Also mention that this webinar is being recorded and will be available for viewing at a later date on the Foundation's YouTube page. So if there are others that are not able to attend, please direct them towards the Foundation Fighting Blindness's YouTube page for more information. So we'll start here with the Translational Research Acceleration Program, or TRAP for short, our sort of vision slide. So why is this program so important? Well, this iteration of the TRAP award is important because people with inherited retinal degenerations basically just need more solutions. Right now, there is one FDA-approved therapy, but there's, with the uniqueness and diversity of this disease indication, we need to be able to provide more solutions for people with IRDs as well as dry age-related macular degeneration. What are we really trying to fund here through these funding mechanisms? We have three major research priority areas. One is the novel medical therapies, genetic technologies, and regenerative medicine. Novel medical therapies cover small molecules and biologics. So if you have a technology or asset in that space, that will be the research priority area in which you apply to. Novel medical therapies also cover sort of the other categories. So if you have something that doesn't necessarily fit within the three priority areas mentioned here, please submit your application under novel medical therapies. And once reviewed, if we feel that it fits better in another category, we will then notify you and then move it into its appropriate group. Next, genetic technologies covers things from viral to non-viral based genetic technologies, as well as gene editing. We'll go into depth more about each of these categories as over the next few slides. The third is regenerative medicine. This really covers stem cells or cellular scaffolds or anything that's really replacing damaged retinal cells for the restoration of vision. One of the unique parts of the TRAP program is that you get access to this proactive management structure, which includes monthly updates, a project mentor, as well as advisory council oversight. And essentially what that means is these wraparound services are really there to help each project continue and accelerate towards the clinic as quickly as possible and mitigating any technical hurdles or any problems along the way and not letting the research slow down. When you come out of this acceleration program through this accelerator, our hope is that the projects are now ready, have been de-risked for engagement with those in the venture capital space, as well as hopefully sparks other partnerships on the journey towards filing an investigational new drug filing, establishment of a clinical trial, and hopefully in years to come, a commercial product for our constituents. Again, this program is all about providing funding and wraparound services to help accelerate a program to the clinic. So what do we mean when we talk about medical therapies as far as one of the research priority areas? We're really talking about the employment of chemistry, biology, and bioengineering to formulate new therapies that mitigate IRDs or dry AMD. So again, the areas of interest would be small molecules, biologics. We're also underneath this category talking about optotherapies. This would include optogenetics, as well as alternative therapies. And again, the alternative therapies is others category. The treatment, we are always looking for agnostic treatments to IRDs and dry AMD. But if you are far advanced and you have a specific drug for a specific disease indication, it needs to be more advanced and sort of almost on the cusp of ready for regulatory submission or clinical trial start. So while we usually want to fund things that have a broad impact, those projects or efforts that are further along will still be considered, but they need to have significant background data accompanied with those. So here are some of the other program attributes, use of high throughput phenotypic drug screening tools, whether they market or target relevant to human and orphan-related retinal degenerative diseases, directly sense and respond to retinal state, and the asset is the ability to prevent further degeneration or restore function when the primary signaling cells are not damaged or non-functional. So these are some of the project attributes. This is not a complete list or not meant to represent that, but these are things that you should highly consider as you're applying for a TRAP award under the Novel Medical Therapies Research Priority Area. Next would be genetic technologies. And we have adapted this image from the FDA just to kind of give you a sense for what genetic technology, what is meant by genetic technologies here for the TRAP award. Again, we're looking for agnostic or even specific to IRD type again, but if it's specific, it really needs to have, it needs to be further along in the preclinical development process. And what we mean by that is deliver targeted genetic material to cells by physical, chemical, or viral methods. And that would be, DNA, protein, lentivirus, AAVs, lipid nanoparticles, everything that's sort of in the tools to actually deliver your therapeutic to the human body that then will augment some part of the genome in order to mitigate any disease phenotype. So that's really what we're talking when we get into genetic technologies. The third and last research priority area is regenerative medicine. And the goal here is the development, regeneration, and employment of human cells, tissues, and cellular or tissue-based products like cellular scaffolds for the restoration of visual or retinal function and vision. Many times these fall in the space of agnostic to IRD type or dry AMD condition. And what we're talking about here is the employment of regenerative medicine techniques to replace damaged cells for the restoration of vision. And not just, so it's very key that we're really talking about the restoration of vision for especially diseases that are far advanced and cellular, such as like photoreceptors are now missing. How do we actually restore visions to these patients through these research efforts. Before we skip here, I see there are 2 questions in the Q&A. I'll just take a pause here for a second and answer those 2 questions. So I think the first question is diabetic retinopathy in scope? Unfortunately, diabetic retinopathy is not in scope currently for the Foundation Fighting Blindness. So we are really squarely focused on inherited retinal diseases as well as dry AMD. The next question is, we are working on a type of LCA that has potentially a small prevalence and smaller market opportunity. Would this be viewed positively or negatively? This would be viewed very positively if you have a great approach to providing a cure for that disease indication. This is what the Foundation Fighting Blindness is all about, is being a platform, a funding source for rare and even ultra rare conditions that have ocular prevalence in the disease landscape. And we are here to help push therapies for these disease indications. So yes, that particular LCA that type that you're talking about is probably well within scope of what we're trying to. Thank you all. Oh, we have one more question. You broadly support small molecule development and delivery modes for AMD. I would say it depends on what you mean by broadly. Now, if we're talking about wet AMD, I think that's a little bit, a bridge a little bit too far for our funding mechanism. But if you're squarely focused on dry AMD, yes, we support small molecule development for dry AMD as long as it's already sort of at a preclinical stage. And we'll talk more about that stage, those stages in a minute when we talk about the different levels of TRAP funding. So let's talk about the TRAP funding mechanisms and criteria. FFB is mostly working in this space when it comes to FFB grants. And these are usually 3-year projects, and the highest level of funding for the TRAP mechanism is $1.5 million. In the next slide, I actually go into the different TRAP funding categories. Again, this supports early preclinical all the way to sort of very late stage preclinical, means IND enabling studies, as well as components of sort of manufacturing and IND submission. So we're funding sort of the whole continuum from early preclinical all the way up into regulatory filing. Again, I'll just reiterate that these projects are really focused on filling knowledge gaps for IRDs as well as dry AMD. And as you think about your therapy, you have to have a great understanding of the potential or current funding landscape. So pay attention to what's currently going on in clinical trials and compare what you're working on to that current state of the art. And does it surpass it, or does it provide a better profile compared to what's already being developed in the clinic? Be able to make that argument. Here in part D of this FFB grants, we say that research should be hypothesis driven. However, if your proposal has already demonstrated that the research plan has already identified a target, is really sort of more in the development stage, the hypothesis may not be required. For example, if you have already shown that what your therapeutic targets, that it is efficacious, that it's safe in a model, and you're looking to do some GMP studies, then you're really more in the development phase, and the hypothesis may not be necessary for that particular type of proposal. But if you're a little bit earlier in the process, it definitely should be hypothesis driven. So don't forget to include that thinking or that language in your application. You also have to be able to demonstrate the technology's potential clinical relevance and the potential marketplace standing. This just gives the reviewers a good understanding for have you completely thought about how your technology will work in the marketplace and in the clinic. And also, and last but not least, please, if you have an asset that's at the preclinical level and you do not have IP protection around it, you really need to start consulting, if you're in academia, the patent lawyers within your institution, or if you're a company, you should really be considering protecting that IP. So that I cannot stress that enough, that it is paramount that you have protection around the asset. The other two, excuse me, the other two funding mechanisms are a little bit less used by the foundation, but can be considered. One is the FFB cost share partnership, and that is where we will maybe, in a matching way, fund up to $500,000 per year over a 3-year funding period with in-kind support. So that is something that can be considered. It's not necessarily traditionally what we want to do. We are really set on running our grants process, which is a competitive process. However, this is another mechanism. A third mechanism is for projects that don't necessarily need funding, but actually need access to our science advisory board or advisory councils for pivoting their technology toward an ocular indication or a retinal disease indication. This is really for companies or people in academia who have been working on something that works maybe in another disease indication, but now have enough maybe background evidence or literature to show that it may work in the eye. So they may not need money, but need the support to fully understand the retinal disease landscape through advice from our advisory panels. So those are the three mechanisms, with the granting portion being the top one. I see we have 4 questions here in the Q&A. I'm going to take a pause here to answer some of these questions. Our first question is, would a non-US company be eligible for applying for this grant? Yes, just period. You will be eligible. The foundation funds all across the world. So therefore, whether it be in academia or companies, you would still be eligible for funding. Could animal models for IRD and dry AMD apply? Yes. If you're looking to develop a unique model and you have for preclinical testing in the IRD or dry AMD space, yes, animal models could be in the funding if that's what you mean. If what you're also asking is, should applicable efficacy for IRDs and dry AMD be used? 100%. It doesn't have to include just animal models. It also can include retinal organoids or other in vitro model systems. But just be ready to back up the use of your model systems for preclinical testing. I keep harping on the preclinical testing. This award is not for basic research. We are really looking to generate data to go into regulatory packages for regulatory approval to start clinical trials. So always think through that lens of, how do we get the technology to regulatory approval and so we can start clinical trials. The next question is, my proposal uses stem cell differentiation as a model for AMD and uses gene editing screen and small molecule screen. So which category would I fall in? Oh, I think this is a difficult one. So you're looking to restore, it looks like, cellular function or retinal function through regenerative medicine, but you probably want to first genetically edit the cells. I think that right now, we'll probably start with the end. This probably should go into regenerative medicine, but we will take into consideration, probably bring other reviewers in to talk about the gene editing portion of that. So I like this question because basically you're spanning 2 of our research priority areas. Submit to 1, but we will ensure that the right reviewers cover all the technical aspects of the technology falling. But right now, since the end therapy is really it seems as if what you're saying here, stem cell regenerative sort of approach, I would fall under the regenerative medicine research priority area. Do you fund NIHPIs and labs? It is our current policy, I believe, not to fund government entities. However, well, I think that I'll just stop there and say it is currently not our policy to fund NIH applications. However, if you have a great idea, I think we would love to see it. So if it wouldn't take too much out of your time to submit a letter of intent, I would do so. I think that it's always great to know what others are thinking, and then, to see if we might not be able to find a way that, the foundation might be able to work with the NIH. It would be something I would like to explore. So please submit a letter of intent. Are PIs allowed to send in 2 letters of intent for the TRAP program? Yes, anyone can submit any idea as long as they need to be different from each other. It can't be the same idea, just a little bit retooled. I would think about, making them very unique to one another. But yes, you can submit multiple letters of intent. Could it be multiple PIs? Yes. We'll go on further in the presentation. We're really flexible with this funding mechanisms. It can be multiple PIs. A PI can work with multiple CROs if that's the case. It really doesn't matter how we bake it. The only thing I will say is that, but we will only be signing award terms with a lead PI. We will not make multiple awards to multiple PIs per application. It would only be one PI awarded, and then that PI will have to set up sub-agreements with the other PIs if awarded. May I see previous 2020 TRAP recipients and their project titles online? You can't see their titles online just yet because we haven't updated our website. We're still working through some award agreements with 2023 award recipients. However, in a later slide, I will show you which TRAP awardees have, some of the TRAP awardees that have been awarded and their project titles. So stay tuned for that information. The next question is, you state that FFB grants are 1.5 million up to 3 years. That's for TRAP 2. So on the next slide, we'll go over the categories. Just to clarify, does this mean to be a cumulative 4.5 million or is it capped at 1.5? It is the latter. You are capped at 1.5 million. And again, we'll go over this in the next slide. Can one team submit 2 separate applications? We have development programs that fall within separate focus areas. Yes, I think I've already answered that in the previous question, so that answer is yes. Next question is, will research only involving animal models be funded? Not necessarily, no. If you have a good, and I'll take a pause here. What we're looking for when in research projects are regulatory, a regulatory relevant approach to your development plan. So, and I'll say that. So again, think about, sort of start at the end. What do you plan on submitting to a regulatory entity in order to start your clinical trial? And work back from there. What data do you need in that package from a preclinical effort? And then start from there. And then the models that you choose, they have to be able to be backed up through literature and sort of preliminary research in order to really be looked at as real preclinical models. So start there. And thank you all for the questions. I love this. They're rolling in. This is exactly what this is for. So please keep the questions coming. Does the foundation try to bring companies together? As an example, if one company has a potential drug but needs a delivery system to get the drug to the retina, maybe not through the TRAP funding mechanism, but if someone highlights that in their application or needs that type of support, we are more than happy to make introductions and to sort of expose developers to our network of people working in the drug development, drug delivery area, so they can form their own partnerships and trying to get that in place. So, so yes, I would say not directly through the TRAP funding mechanism, but it is part of our mission to ensure that people that we fund have all the resources at their fingertips in order to help, bring along their technology. But it wouldn't necessarily come directly through sort of a TRAP funding award. But yeah. Please, if you need help, please reach out to me or anyone at the foundation so we can help start that conversation. I think the next question, must the grant be spent in the US? No, it can be spent abroad as well. I think we have funding throughout the world, especially we're funding a lot of projects in Europe and the money is spent over there. So no, the money doesn't have to be spent just in the US. Is preliminary data needed? Yes, you have to have preliminary data. Again, the TRAP program is about advancing an asset or advancing an actual technology that exists. And that technology needs to fall in those 3 research priority areas. It cannot just be an idea. You have to have actually something tangible with some level of background information that it will be beneficial to treating inherited retinal diseases, and or dry AMD for us to fund that type of project. Would FFB fund multiple programs for the same exact target? Yes. So I think the best example is we're funding many different projects that are targeting Stargardt's disease. So yes, but they all are taking different approaches to how to mitigate the impacts of Stargardt. So yes, we will continue to fund programs if something is clinically available for the disease indications that we're most concerned about. I'm going to take a pause here from the questions, so I'll get to them. So please hold on. I see there's 3 more, but let me get through a couple more slides and then come back to the questions. Here are the TRAP funding categories. We have TRAP 1, TRAP 2, and TRAP 3. And essentially, if you think about the full sort of trajectory of what preclinical testing is, TRAP 1 really covers early preclinical development from target validation to proof of concept and up through, you developing your therapeutic lead. So if you're thinking that, well, I'm very, very early on, but I have something in hand that I need to validate that it targets, this biological target, and then I'm going to, use the funds to maybe help start creating a lead therapeutic, that's what TRAP 1 is for. That is capped at $900,000. And what we're looking at is funding that over a 3-year period. So usually we're maxed out at giving you $300,000 per year over a 3-year period. Again, capped at $900,000. Now, with the TRAP being having that word acceleration in it, if you think you can execute your program in 2 years or 1 year, then let that be known. It doesn't have to be spent over 3 years, but for budgeting circumstances, this is how we're currently budgeting and rolling it out. But you can put a shorter timeline in if you so choose. TRAP 2 funds late-stage research that covers lead optimization, again, sort of picking up where TRAP 1 ends, through drug candidate selection and IND-enabling studies. So we're really talking about phase 2 going all the way up into and hopefully through IND-enabling and hopefully even IND submission. TRAP 1 is a special category. This one has the highest bar getting approved, but it's really a flexible funding award mechanism for very, very late stage preclinical efforts that are focused on getting someone over a sort of transition hurdle. Let me give you a good example. You may be trying to file an FDA IND filing. You may be talking with someone in the venture capital space that says, hey, it would be great if you had this one experiment before we can invest. That's what TRAP 3 is about, is an infusion of up to $1 million, no time limit, to really do that 1 or 2 critical experiments in order for the next natural transition point for that effort. Again, all focused on IRDs and dry AMD. Eligibility criteria. Again, I think I've sort of spoken to that over the time, but again, it has to align with the FFB or the foundation's mission of IRD and dry AMD therapeutic development. You need to really understand the models that you're using. The models have to be favorable for regulatory filing. We don't want to use models that aren't going to help us get to it. And then you must choose your funding category, and then also your research priority area. And here are some of the TRAP applicants must meet the following criteria. The person applying must hold a leadership position from the organization that you're applying. You have to be able to submit, receive, and execute the dollars that you're asking for. Again, each effort will have a single principal investigator who is responsible for the award, and that covers project oversight, financial management, and reporting. So essentially, who is the person between the award and the foundation who is going to be responsible? Now, you can have multiple sort of co-PIs or sub-awardees, but that will be on the principal investigator and their organization to set up those sub-awards. You must be able to fully demonstrate that you have the research team and environment able to execute what you have. Again, you can work with the CRO, but you can't come in and say, I'll work with some CRO, but I don't know who they'll be yet. No, you have had to already vetted them that they have the capabilities to do the work that you're asking about. So you have to fully demonstrate that the award will be used along the lines of the mission of the foundation, and as well as the environment and the personnel have the skills and the equipment and the resources in order to execute the award and the research plan. And again, it sort of speaks to having all of that stuff established. You know, we say any proposed research partnership must be established. They cannot be notional. Have these conversations now while you're putting together your letter of intent. Have the conversation about if you're going to partner with someone, if you're going to hire a CRO or contract with the CRO in order to conduct your research. Have those conversations now so you can understand what that relationship will look like and how much money it will take in order to formalize that partnership. This is a new slide that we've added to this deck. So we're in the 4th year of doing these proposal days. But one thing we want to make it clear is to companies that apply for the TRAP award, you know, no matter where you're at in the world, if you are invited for a full application, there will be basically a company due diligence meeting that will need to be set up in addition to applying to the TRAP award. And the reason being is that we want to make sure that before we award a company, that the company is healthy. So we'll look for information such as, we'll analyze the capitalization of the company, look at your revenue, profit, and margin trends right now, who are your competitors in the space and in the different industries, and who manages and sort of owns the company. The Retinal Degeneration Fund, which is a subsidiary of the foundation, will be involved in this process, and it will require a pitch deck for it. So this slide is just to give companies a heads up that if you are invited to submit a full application, you will be required, in addition to the full application, in order to, you'll also be required to participate in this meeting. It gives basically a nice overview of the application or the selection process. Right now we are, on May 12th, for the proposers day. Letters of intent are due June 1st. Once those are reviewed, the full application submission request will be sent out to those who are being invited by August 10th. We will then start the full proposal review, which runs between October and December of 2023. And then we go through our selection and notification process, which will conclude around March 2024. I understand that this is a very long process, but we think that, for this level of funding, especially from the foundation, requires the time and effort from our review committees to really vet these projects and make sure that we're funding the best and most promising projects through the TRAP award. Once selection is noticed, award terms will be sent over. We will then have a kickoff meeting, and then the research will start. For someone who asked who were the 2023 awardees, this is actually the first public notice, and we have made 5 awards. We're still finalizing a final award, so that particular award is not being publicly mentioned yet, but the 4 who we do have award terms with are Dr. Catherine Peppel, Dr. Renee Rouse, Dr. Barbara Rohr, who is representing a company who we have funded called Mitachem, and Dr. Christoph Palczewski. All are 2023, they were awarded in this particular application cycle. So you all will be applying for the 2024 award. So if you know these individuals, please congratulate them. We are very excited about the projects there, about their TRAP projects. And just for just a little bit of an overview, we are running a little bit behind time, but I'll give it a second here that Dr. Pepley, excuse me, I think I'm pronouncing the name wrong, has been awarded to really find a intervention for sort of AAV-induced ocular inflammation. We all know that AAVs are sort of leading the field when it comes to gene therapy delivery modality. However, they do inherently cause some level of ocular inflammation. We want to put research in place where we can actually mitigate that process, so we can really take advantage of AAV's utility in delivering genetic material for therapeutic use. Dr. Riles is looking to deliver CRISPR prime editing tools using lipid nanoparticles to actually correct mutations in the perforin 2 gene. So we're really looking forward to that one. Dr. Palczewski taking a similar approach, but using twin prime editing to maybe try to create a platform to actually address mutations in inherited retinal disease causing genes, and create a platform to address many, many different genes. And Dr. Rohr is really taking a neuroprotective approach for dry AMD. So this should really, this targeting the fuse protein, we're looking to see if it can mitigate some of the retinal degeneration caused during macular degeneration. An additional layer for the program, I mentioned that you also get access to a project mentor, as well as the TRAP Advisory Council. And the TRAP Advisory Council really is just overseeing the projects, but in a sort of, in not an ad hoc, but just every few times a year, this advisory council. And they are really giving, in general, instead of reading through all these things, just different levels, whether it be business advice, industry advice, engineering advice, regulatory advice. You know, we bring these individuals in to give us an overview of where the project stands and their expertise and how to move it forward from all of those different angles. Those individuals that currently sit on our TRAP Advisory Council are listed here. We have Dr. Nima Mayhew, Ash Deragopal, we have Jared Cheatham, Hema Ramkumar, Mark Fields, and George McGrath. So I'll publicly thank them for their continued service, but they're really over here to just look at the landscape of each project and provide their expertise on how to more quickly move these projects towards the clinic. Again, this is the end of the slides for the TRAP overview, and I'll just leave this up here so you can pay attention to our key goals, our ultimate goals, and then key dates that you need to pay attention to for the application process. I'll take a pause here and again, dig into the questions that we have here in the slide. Here it says, would it be suitable for the development of a device that enables specifically innovative research on retinal degeneration? I'm not 100% sure what you're asking here, but in general, we don't fund the development of devices. We're really looking for sort of more biological-based solutions for our disease indications. If I didn't answer that question the way you mentioned, can you please maybe ask it in a different way that so I can try to get at your question? Can we get a copy of your PowerPoint presentation from your webinar? Of course you can. Again, I'll say that I can provide PDF versions of this PowerPoint so you can more easily peruse through the things that I covered. Also, again, this webinar will be recorded and available on our YouTube page for viewing at a later time. Okay, next question is, can you please share examples of robust preliminary data submitted by your former applicants that helped them secure TRAP2 funding? I would have to ask that sort of the preliminary data that they shared. I would have to ask the applicants if they're comfortable with me sharing that data. If you could please reach out to me directly, I'll share my email at the end, and maybe we can actually get you linked up with a current TRAP awardee, so maybe they can more personally maybe mentor you on the application process. A lot of the things that are a part of the TRAP award are confidential, so I don't want to share any information that a particular awardee would not want me to. So I can, please reach out to me by email, and then I can find someone maybe that can help mentor you or share their process with you. For gene therapy, we have in vitro data, and next we want to do in vivo studies and progress to IND. Is just in vitro data sufficient for TRAP2? I believe so. If you've already shown in vitro data and you're looking to actually do animal studies at this time, I believe that is, but make sure that you clearly show what your data is showing, and then justify why you believe it is at the level of TRAP2. Next question is, if we believe we might qualify for, say, 2 of the TRAP categories, are we able to speak to you to gain FFB opinions as to which category is more relevant? Yes, please reach out again through email, and we can set up a quick call to discuss your project. all I ask is maybe you just send me a brief summary of what you're doing so I can be prepared for that meeting. Again, it's not like the NIH, once the solicitation is out, you're not able to ask those types of questions. Please reach out to me. I have no problem with having a quick conversation or even a quick email exchange to maybe clarify things. In the online application, there's a place for attaching the CV. Can we include CVs of PIs and co-PIs in the field attached? I believe so, if you plan on, if it's a multi-investigator approach. If not, and if you're having struggling, please let me know. We have our grants manager online as well, Leala Kinney, and she can help us figure out a solution to that problem. Does large-scale data screening belong to Trap 1? Yes. Yes. I'll just say yes, because if you have not really identified your tool compound at that point, or maybe if you even have a tool compound that you are expanding from to find something that's, let's say tool compound or lead gene therapy, and you're looking to expand it to make it more efficacious or safer, then yes, I think it belongs in Trap 1. If work involves a CRO, do we need to have scope of work in quotes already defined and worked out for the letter of intent? No, but I think that you need to notionally know how much it's going to cost. So if the CRO can give you a ballpark figure of how much the work is going to cost, you will need to include that in the letter of intent. There is a small section of letter of intent where you talk about budget, but we'll need to know a rough estimate. So if you can try to figure it out, that will help your letter of intent provide value and understanding around that. Do you expect to fund a similar number of projects as in the 2023 cycle? No, actually, we would like to fund more. So yes, I think that in this next cycle, in full transparency, I would love to fund 7 or 8 projects in this next cycle. Can an NIH researcher be a co-investigator in an application? I would say that we would, and I'm studying around this because our current policy is not to fund NIH research again. But again, we would love to see the proposal. We would love to understand, and I think we could spark up another conversation about that. But currently, if I just had to say where we currently stand, no, NIH research cannot be a co-investigator unless it is a non-funding partnership at this time. FFB funds are not meant to go to fund the NIH or National Eye Institute for that matter. However, it could be discussed. Early stage companies may not have deep funding, no revenue. So is there any minimal bar? I'm not fully understanding the question, but if you're saying that basically your startup company has not been capitalized yet, that's fine. I think that that's fine. We fully understand that if a company may be just now spinning out of an academic lab and has not capitalized at this point, that's fine. I think that one of the key criteria, though, is that the company has worked some deal to actually acquire the technology from a legal standpoint, whether an official licensing or something of that matter. It often muddles the water when a technology is still in a university setting and the company is trying to be stood up. But it is not, we work with plenty of companies that are just starting off. Who owns the resulting IP? Host institution or FFB or both? The host institution owns the IP, period. In our award terms, there will be some level of revenue sharing that is a part of our award terms, but the IP is fully fully owned by the applicant. How many Trap 1 awards funded in 2023? I believe there were, of the 4, I believe there's 3 funded in 2023. And I'll say that we don't necessarily, we're funding the best research. So I wouldn't worry about how many Trap 1s were awarded versus how many Trap 2s. We're going to fund the best research that has the most promise of getting to the clinic, or we can have the most promise of accelerating towards the next logical stage of preclinical development. That is our main goal. So we are going to look at our budget. We are going to see which awards have the main thing, have the main basically are the most promising that we can help move forward. Photobiomodulation using far red to near light has been shown to mitigate retinal disease. Are we eligible to apply for TRAP funding using this technology? I would say yes. Please put in I'm just getting a 100% confirmation from our VP's of Sciences Award from Amy Laster, and that's a resounding yes. So yes, please submit a letter of intent for that particular approach. Our lab was funded by FFB from 2004 to 2013 to study light therapy. I think this is a similar question. Can we apply for TRAP funds to move this in the clinic? Yes. Yes. So I'll answer, say, oh, same person. Yes, we can. Can we apply with a current unfunded FFB IRA grant? Peter, interesting question. I believe so. So are you saying that you've submitted for an IRA grant? It's probably currently under review, but you want to submit a similar idea for the TRAP award? Yes. I think that until you're funded through any of our mechanisms, you are more than free to apply for any of our funding mechanisms that you're eligible for. Can you define revenue sharing? No salary for PI, is that revenue sharing? No, when I'm saying revenue sharing, it would be more so if a company is coming in and or any entity and saying they want to advance a company, but they're going to also throw in money themselves that they have raised, but they're looking at basically, for example, we need to do GMP or GLP safety studies at a CRO. It's going to cost $2 million. A company only has currently $1 million, but they need another $1 million for the FFB to help do the entire experiment. That would be an example of revenue sharing for that particular research effort. So there could still be salary for the PI, but the revenue share is more so that a single entity does not have the funding required in order to advance the technology through a particular research activity. So we're going to share the cost of the event. Could you elaborate on the revenue sharing items? I think this could be a showstopper for some applicants. Okay, let me make this clear. There is no requirement if you're applying for an FFB grant to revenue share. There is no requirement. The revenue sharing aspect of it is just another category. The FFB, and actually, I'm going to go back to that slide for everyone because I want to make this clear. The FFB grants here is what we're mainly talking about. This is our competitive granting process. You do not have to bring any money to the table in order to apply for this whatsoever. This will be what the vast majority of people who are applying for TRAP awards are in. These other two categories are things that we can also consider as other relationships, but these are not the tried and true ways in which we are providing funding to applicants. It is only through the FFB grants, and these require no revenue sharing. Next question is, if applying from outside the US, is the US collaborator essential? No, it is not required. You can have all work can be done outside the US without any US collaborator. If an organization trapped to funding, can they apply for TRAP 1 at a later date? I'm not sure about the question. I would say this: if you're not sure about which category you should apply to, whether TRAP 2 or TRAP 1, please send me an email so we can have a follow-up conversation about which category best suits your research plan. I'm sorry. Okay, so just give me 1 second, audience, so I can maybe try to fully understand this question. If you receive TRAP 2 funding, can you then apply for TRAP 1 at a later date? not for the same project. So if we're funding the acceleration, we would want you to move forward in that funding, especially if it is successful. Now, could you reapply once that effort ends for Trap 3 funding? Definitely, especially if there's one or more critical experiments that need to be done in order to help the project move towards the clinic. Next question is there a specific detail of what needs to be in the letter of intent? Yes, if you go onto our website, and Leila, if you're still there, if there's any way you could put that in the chat or in the Q&A, or send it to the participants, the actual link to the letter of intent or to the TRAP's webpage, you can it provides all the details there.
I just put it in the chat.
Okay, so Liala's going to throw the link into the chat that will give you what needs to be in the letter of intent. We have guidance there. Liala, also, let me see. Let's see here. I see that people have also populated the chat with questions. Liala's asked if you would, I'm going to try to answer some from here, but if you need to ask a question, please put it in the Q&A box. I guess we didn't cover that before the conversation started, but we're tracking all the questions in the Q&A feature. So if you could please put your questions there, I will try to get to them there. I think I've answered that one. Do these apply to human research or can be animal models? I think that most of our TRAP projects apply only to animal models since we're talking about preclinical development. However, if you're including human-derived iPSCs or retinal organoids as human research, then yes, TRAP funding would also cover that as well. I think I've answered that one. I'm just going through the chat right now for everyone. Light. We have a question here is, if we have a therapeutic ASO with preclinical plus data, can you help link to a manufacturer, or is that too specific of a question? No, I mean, again, we're happy to introduce you all to potential manufacturers, but it's really on you to vet those individuals who you think would be best suited for your technology. So, but if you are awarded a TRAP award, I would have a list of people that you've been considering as manufacturers, or even now in the generation of your letter of intent, if you need more advice about who might be a great manufacturer, please reach out to me, and maybe I can hook you up with a mentor in our field that can maybe provide some guidance to you. All right, so I think we have about 7 minutes left. Again, keep the questions going. I'm going to go down to, again, we've stopped here to really highlight the ultimate goals and the key dates of the TRAP award. But again, I'm going to kind of foot stomp my thing, my position on grant writing tips for the audience. Some of you are seasoned veterans when it comes to grant writing, and others aren't. I want to make it clear through providing these sort of bullet points here that, please take some time and effort in polishing and refining your letter of intent. And if you're invited for a full application, your full application as well. Meticulously go through it, looking for grammatical errors, making sure that the data that you're providing may be in graphic form, is clear and easy to understand, and also aligns with the narrative or the story that you're presenting in your application materials. It's very important. The tips I'm gonna give here, I'm just gonna go through and read them. I think it's just easy. And please, if you have any questions, please throw them in the Q&A part, the Q&A feature of the webinar. Really start with a clear and concise project plan. And essentially it's like, what are you attempting to do with your technology? What disease indication are you targeting? Clearly outline your research methods and your plans. What are your expected outcomes, and what is the potential impact of the project? Clearly state that, and that can be done within a few sentences to really help orient us as well as the reviewers to what you're actually trying to do. Get to what you're trying to actually what you're trying to do up front. Make sure that your proposal aligns with the goals of the foundation. I think I've highlighted these a number of times. We are squarely focused at this current time on inherited retinal disease and dry AMD. We will not be considering other disease indications as of right now. So go back to our website, look at the TRAP application materials. I think it overly emphasizes that our research priority areas and are the disease indications that we care about. Seek feedback from mentors and colleagues. Again, if don't do this in a silo. Really take the time to ask people if there are questions, to help refine your research plan. Again, we're here as a resource as well. If you're looking, if you have great technology, but you're not sure how you're going to deliver it, reach out to us. Maybe we can introduce you to some people that you can form a partnership with that can help solve that problem for you. Highlight your team's expertise and qualification. There may be some of you that are new to the retinal disease space, but you have a really promising technology. Really, in a very concise and direct way, highlight the expertise and qualifications of the team that you're formulating for a TRAP submission. It is very important that the reviewers, if they don't know you, that you show that you possess the technical and maybe even from company standpoint, business expertise in order to move this technology forward. Do not shy away from showing the potential challenges of your research plan. This goes a long way with reviewers. If you have thought intimately about your research plan, there are going to be challenges that may arise during the research project. That's fine. Highlight them, propose potential solutions to them. You have a particular way you want to execute your research plan, but things may go left. You may have to pivot. Mention those and just be upfront about that. The last three that I'll mention here is, please emphasize the significance of your research. And if you are successful, what are the logical next steps? We're putting, from a foundation standpoint, we're putting a lot of money behind these projects. So we want to know that you're thinking years in advance, especially if you're successful, how you plan on getting this to the clinic and in what way. Again, I cannot emphasize more that you have to use clear and concise language and avoid using jargon when possible. Make the application very easy to read and tell a story about your research plan and how you plan on, mitigating one or many of the disease indications that we care about. And again, please proofread and edit your proposal carefully. Seeing typos or mistakes, it really is, it dampens a reviewer's enthusiasm for your particular proposal. All right, well, we have about 2 minutes left. I don't see any more questions in the Q&A. Leala has actually put my email address in the chat. So if you have any further questions or would like a follow-up meeting, please reach out to me at that address and we'll try to get something set up. I see that there's another probably question. Jasmine, thank you so much. And I'll thank the entire, I believe at one point we had 70 people in attendance. I think that's the highest number yet we've had from these proposers days. I would like to thank all of you for joining today and taking time out of your day. Again, if you have any questions, please do not hesitate to reach out to me. Here's my email address here. Again, we will try to send out a PDF of this of the presentation, as well as the recording will be on YouTube at some point in time for you all for viewing later. We are really, really excited about this year's award process. Please, submit your letters of intent. If you have any questions, again, reach out to me. More than happy to put you in contact with others that might be able to help you during your process. So have about a minute left. Are there any last questions? I'll maybe pause here for a second to see. Seeing no other questions, again, I'll just thank you all for your time and looking forward to your submissions. Take care.